Test, test, test.
Engage. Welcome to the September meeting of the Silicon Valley Fourth Interest Group. I was on the verge of putting out the call for participation to the fourth day, which is coming in November, but got hung up on not having done any legwork or preparation for sending that. So it will appear shortly. It shouldn't constrain you. It shouldn't inhibit your planning or preparing for fourth day. Andreas is going to go get Ting, hopefully, who is just ringing the doorbell as we speak. Well, Andreas is going there, and we'll see what happens. In the meantime, I wanted to say coming next month is the exciting history of Ampex. I don't know, as many of you are aware that they took down the Ampex historical sign. I drove by the process, working in Foster City and living in Sunnyvale, so I would drive by there every morning. So we'll have a moment of silence and a nice talk about the history of Ampex, and we might talk also about other things that have left us, like Orchard Supply. Halted is leaving in January. If you haven't made it to Halted Specialties, I recommend it to you. Let's see, Orchard Supply, Halted, Weird Stuff Warehouse, the list goes on and on. Ting, do you have slides and computer stuff?
You don't look forgot to get my computer into my car.
Would you like to?
I can talk. Okay.
I know, and I've always been very grateful. So we'll when you're ready. When you're ready, I'll stop blathering and you can get up here and go. You ready now?
Let me get a cup of coffee.
All right, Ting's going to get a cup of coffee, and I'm going to blather a little more. We were talking about things that have left us. Anybody else? Orchard Supply, Weird Stuff Warehouse, Halted. It seems like there are a couple of others that came to mind when my mind was working, which isn't this morning apparently. So one thing I do during fourth day that doesn't happen during the rest of the year is I keep careful time. So I was thinking about things I could do to have an active schedule, like a spreadsheet of starts and stops, and it would count down each speaker and, you know, issue flashing lights or some cues that say, okay, wrap it up, you've got 2 minutes. Maybe in 2 parts, I was thinking of having like a schedule that you download to a computer that like a Raspberry Pi or something that would have a screen that is off to the side that would serve as the timekeeping display. And then I thought, oh, maybe not so smart. Why don't we just have all of that stuff running on the laptop and then put it out on the web, on the internet, over Wi-Fi to a Raspberry Pi that's just got a web browser running for that particular page. And that seemed like a more straightforward approach to that problem. The other, and then I could give a talk about it. The other The other thinking that I was thinking for a presentation that isn't really fourth is to talk about the technology that allowed us to win World War II. There's a very good book called The Wizard War. And relatively recently, last year or so, I ran across another book called The Deadly Fuse. And if you could get your hands on either of these tomes, which are now, of course, out of print, I recommend them to you. But if it hadn't been for technology, pretty clearly we would have lost World War II. And in my opinion, the biggest factor is one that 99 people out of 100 on the street are completely oblivious to. This idea of the proximity fuse, I feel, was a really big factor in that war. And without further ado, we're going to have a complete change of subject. A man who needs no introduction.
All right, let me just close this one off so I can use the board. Yeah, I think probably need some more. Okay, I'm C.H. King, and unfortunately, I was in a rush getting out of my house. I got my computer sitting at the door to my kitchen. And I ran out to the car and pick up lots of things that a friend wanted me to bring. And I forgot to take my computer with me. But since I was working on this subject very intensely in the last month, I think I can remember most of the things that I want to talk about. And last night I was working very intensely on the PowerPoints. So I still think I can remember most of the points that are in the slides. But I'll give you, Dave a copy of the slides. So if you are interested, you can always have it later. So what the topic here is exhaustive pattern search. In bacteria. Genomes. Okay, so this is the title. And the reason of this was that I was fairly focused on microRNAs. And I believe that microRNA is the key to the working of the cell computer. And I would make the hypothesis that microRNA is the basic instructions in a cell computer. So as we know, I think every cell is a computer, okay? The evidence is that it has all the code in DNA. Every cell has one or many copies of the DNA. And the DNA, that's a read-only memory. Yeah, that's, I think, for some reason, DNA seems to be much more efficient than the silicon read-only memory. But the DNA has all the information that the cell used to perform its all its functions. So that we have the DNA. And the information in DNA, how do a cell use the information? And it copies useful information into RNA. And then the RNA would do all the work. And there are certain editings information going from the DNA to RNA. Sometimes the information gets edited. So the NRA is pretty much like a RAM memory in a cell, in a computer. Yes?
RNA is done its thing. Does RNA dissipate? Does it go away? Is it used once for a task, or does it set up a factory?
The RNA has a short memory, lifetime. Okay, so RNA somehow it performed its work and then it disappeared. Okay, so it has to if the same function must be done again, new copies of RNA must be made. So there's lots of different RNA molecules, and the most important, or the one that we understand the most, is called the mRNA, which is called a messenger RNA. And each messenger RNA is used to produce one protein. Okay, so there are lots of proteins being produced in a cell, thousands, maybe tens of thousands. And each protein, if it has to be produced, the information has to be transcribed from DNA over into messenger RNA, and the cell has a mechanism to synthesize DNA, synthesize a protein molecule from the information transcribed into the mRNA. This is what we understand. What the information which gets transcribed from DNA to mRNA are called coding DNA. Okay, so this part of the DNA, I think this is the biologist has understand most. But coding RNA, for very low, very simple form of organism, the coding RNA seems to occupy lots of space in the DNA. But for higher form of creatures, the portion of coding RNA gets smaller and smaller. So in human being, the coding RNA is only 1.5% of all the RNA. And the rest of it used to call junk DNA, because people don't know what they are doing, and they feel it's just junk. Okay. Yes, there's also all different theories about the junk RNA. But lately, people changed the term. They found that these RNAs are not junk. There are functions in them. What function? Well, we don't understand completely. But mRNA, we do understand, and the part which, and besides mRNA, people found lots of different DNAs which are not which are not messenger RNA, which are not used to produce protein. There's a whole bunch of them, and so many of them they call the non-coding RNA. And among the non-coding RNA, they found a lot of microRNAs. And the microRNAs interesting is it has 20 to 24 bases. Okay, so they are really small pieces. and they found that originally they thought, well, it's that when they break up a cell and extract DNA, RNA, or this or protein from the cell, and they might just have accidentally breaking the RNA molecules, and so you got lots of bits and pieces there. But then the people found that microRNAs have specific functions. And one of the very important functions is that it controls the expression of mRNA. So certain microRNA is actually activating or deactivating messenger RNA. So that becomes, and I think microRNA being molecules with 20 to 24 bases, it is very, it could be the instruction set. It might be representing the instruction set in the DNA, which controls all the other functions. So in the last couple of months, I was talking about microRNA and what I think microRNA are actually doing. So with this this number of bases, each base, this is the contains 40 to 48 bits. So each of microRNA contains 40 to 48 bits of information. And that has the very nice specificities to encode different functions in the cell. So I was postulate that the microRNAs are actually the instruction set of a cell computer. So, and I found that there's a lot of microRNA clusters. In many of the non-coding RNA and even in DNA. So these microRNAs tend to form clusters. Okay, and this is very nice in that these clusters might be high-level instructions of a cell. Okay, so we have lots of microRNAs which causes the generation of messenger RNA to produce protein. And it also causes the produce of many non-coding RNA, which performs other functions which have yet to be identified. And the microRNAs forms clusters. And this is just like a fourth high-level instruction. And in fourth high-level instruction is usually represented by a list of instructions. So I think the existence of the clusters of microRNA very likely representing high-level instructions in the cell, and thus they are represented as lists. So the one microRNA may cause the produce of an RNA which has a list of microRNA, and these microRNAs are released, and these microRNAs may cause the formation of messenger RNA or other coding, non-coding RNA. And this, just like a fourth computer, we have low-level instructions which produces, orders the physical components in the computer to perform and data operations, and but the bulk of a fourth programs contains lists, and I think using the same principle, the cell may use microRNAs as the instruction set, and then the clusters of microRNA, they are lists of instructions, they are high-level instructions, and the one microRNA can call a list to release a sequence of microRNA, and this sequence of microRNA would then go out to produce other RNA molecules which performs or which can perform very complicated functions. Okay, this is my thinking, but lots of microRNA clusters in the non-coding RNAs of many species. So I was thinking, well, you know, the higher life DNAs are just too complicated. So I try to focus on the bacteria. And the bacteria generally has a much simpler DNA, and I can handle them much more conveniently in my computer. So at one point, I took the entire list of microRNAs that I've collected, and then go on compare these microRNAs with the DNA, with the genomes, with the bacterial genomes. And I couldn't find any match. You know, when I compare my list of microRNAs against higher, the genomes are higher life forms, I usually can find some many clusters. But when I do the same thing with the bacteria, no match. Okay, so the bacteria that I have studied, let me just try to list them. In my slide, I have their official names, but I just couldn't spell them. I couldn't remember all this, so I can only remember a few of these. One of the Nashua and Genitalium and Equidens and the I called acetyl and E. coli. As I remember, this one has 120 kb, not that the kb, that's base. I think this one has 1 kilobase, and Genitalium, that has about 400 kb, and this has 500 kb, and acetyl, that has 2 million, and E. coli has 4.6 million bases. So these are the set of bacteria that I studied, and these two are not actually bacteria. They are called archaea, and their genomes are not complete, so they cannot survive by themselves. They have to be in a host. Okay. Yeah. 你知道我的啊 Okay, so these two has are the they has to symbiotic. So they have to live in a host close to a host and rely upon the host cell to that they cannot produce themselves. And therefore they have their genome size is much smaller because they do not have to code lots of essential proteins. But these two are the smallest bacteria that can self-sustaining. So they can live by themselves, but they have to be live well with they are parasites. Okay, they have to live on the host, but they can survive without a host. And this is the bacteria which produce ferment lactose, which produces lactose. So that's where the German Kraft was made. They would produce lactic acid from cabbage. Okay, and E. coli, of course, everybody knows that it generally lives in our lower intestines. So these are the number of bacteria that I studied, but they do not compare their genome with the microRNA, and does not have any match. And so I thought, well, there's got to be, if my hypothesis is correct, there must be something in the bacteria which performs similar to microRNA. And because the most basic cell function, this is to survive, to divide, and to ingest nutrition, convert nutrition into body parts. So these very basic functions are shared among all the cells, and if they perform same function, I expect they have the similar instruction structure of a computer. So I said, okay, I can't find any microRNA, but I would like to know if there are 20 base patterns. There are repeated 20 base patterns in this RNA, and these are candidates for something like a microRNA. So if I can, you can find, identify unique but repeated patterns, and then I would look for clusters of them. If I can still find clusters of them, maybe they have the same type of functions, but it's just that these base patterns are not the same as microRNAs in high order organisms. But if they use the same basic basic computer architecture, I should be able to find 20 base pattern or larger bases, which has similar functions that I found in the other higher organism. So the problem is to identify all 20 or repeated 20 base patterns. And this is what I tried to do last month. I think I started months before, but then when I move from the simpler ones to the more complicated ones, especially in E. coli, when I have to search 4.6 mega bases for patterns, it becomes very it will take a very long time. And so what I try to do is so I have to use better search algorithms, which are especially tailored to very large genomes. And now I was able to the yeast. I think the yeast is 12 mega. And I was even able to do the yeast and then the nematode. Nematode is the simplest animal, okay. And the nematode is 99 megabase. So that's quite large. And in human, the chromosomes in human are generally 200 megabyte, and we have 23 of them. Okay, so I need an algorithm which can handle this size of genome. So I think last month I talked about I have started doing it. So what I did was take 200 links. I would break down the data file into 256 links. Okay, each link start with four bases: AAAA, then AAA, C, AAA, T, to T. So each base is equivalent to 2 bits of information. So 4 bases has 8 bits. And as you see, 8-bit pattern, there are 256 different 4-base patterns. And I would construct 250 link lists and using search the entire base for AAAA, for consecutive AAAA patterns. So in each link, I would record the distance from one AAAA pattern to the next one. So I just record that distance, and then keep on put all these links together into one list. And this would identify all the occurrences of AAAA pattern. Okay, and the distance between 2 patterns are encoded by consecutive bytes, and each byte have the So if the most significant bit, this is the continuation bit. So if this bit is at 1, then the next bit, next byte is valid. It is with it. So you have 0 to 127, and the next one, if it contains the next 7 bits. And so when the last byte has a 0 in it, and I say, okay, that's it. I get all the bytes with the 1 most significant set has only 7 bits, and when the most significant bit is cleared, and this is the last 7 bit. So this is the most to encode distances among different patterns. So I use this one to break the entire data file into 256 links. And then if I want to search for a certain pattern, which begins with AAAA, then I only have to search this link. And this will reduce the search time by a factor of 256. So that's very dramatic, okay? And earlier, if I just use the simple method of searching, it took 10 days to process this file. And when you use this one, it's reduced to about half an hour. So it's very efficient because you just reduce the search time by a factor of 256. But when I try to do the yeast, it's getting more still with this algorithm. Yeast, I can barely do it, and nematode is impossible. I think the one I use, it has been working on the nematode for 20 days and still haven't finished yet. So I think this is not good enough. So what I did is to expand it to 124 links. So instead of 4 5 base patterns, use 5 bases to form the links, and of course it increased the searching speed by another factor of 4. So I think that's I can still do for bigger ones. I can still expand the links. There's no problem, but I think I was only focusing on the genomes, so that is not that problem at this moment. So to search, identify all unique 20 base patterns, okay, in each and every one of these genomes. And let me see. For this guy, I think I found 146 repeated pattern. So I think the strategy is that the first time I go through it, I just started start each and every link. I have the first 20 base pattern, and I use this 20-bit pattern and then compare with the next next. So here in this linked list, I have this one and a distance and another AAAA, and then so I got lots of AAAA patterns all linked into a list. So I want to find out in this list all the repeated patterns beginning with AA and AA. So what I did is to take the next 16 bases and compare with the next 16 bases. If it doesn't match, I go to the next one, and I go to the next one, and I go to the next one. Okay. When I hit a match, in this list, if this first one has a match, then I just record the address of this one, and that's a match. Okay, it may happen many more times. All I want to do is identify unique 20 base. Okay. In this first run, I just try to identify all the unique but repeated pattern. And once I got this file, I got this many patterns, but there are lots of redundancies in it. So if imagine I have three patterns are the same. So the first time I record this one, and then I scan down here. This one, I found this one was a match, so I record this one. But then when I found this one, it can have another match. So this one was also recorded, but they are the same pattern. So all these things has to be all the repeated need to be taken out. I wanted to find unique 20 base pattern. So I have to trim that pattern list and delete all the repeats. And one of the most serious problem is that in more complicated genomes, there are the M messenger, the messenger RNA are repeated. So there are lots of repeats protein. And whenever this if two patterns are repeated, if there are two repeated protein, and they are the same, and if they are the same, it would, for example, if this is a 2000 base, then the first 20 base match, right? But next one also match, and this also match. So the pattern has a displacement of 1. So it will repeat 2000 times, and this has to be taken out. Okay. So what I did was in this list, and I order them by the address and look for a displacement of 1 or 2 or 3. And I set a limit that I'll eliminate all the patterns with displacement less than a certain amount. And for the simple ones, I think I just set up the limit as 10. And for the more complicated one, I set the limit as 20. Okay. So this will throw away 80 to 9% of the repeated pattern that I can identify. And so then the rest of it, after I threw away these, I would look at the entire list, and so I have address, and I have a pattern. And if I use this ACGT pattern, it's the 20-bit pattern, very difficult to look at. Okay, you cannot visually identify them. So each pattern, I'll give it an ID. So the ID, I think the first one I found about 300. Of these patterns, 300 are spaced wider than 20 bases. And so I would give them unique IDs. So from 1 to 300. And then I can, so these patterns, then I also listed the displacement. So the displacement, if they are totally unrelated, you have a couple thousand bases distance between different patterns. And if these things are only about 100 or less, then you can see, I can visually see the clusters. So I was going through all this list and look at the displacement to identify clusters. So there are lots of clusters I can identify this way. But when I first started, I wasn't even with this displacement, eliminate all the redundant, the repeated protein pattern. I still find long list of clusters, even 100 of these. But then I found many of these very long clusters repeated in the genome. So that indicates these are again proteins which may not be consecutive all the way, but they are consecutive to certain length. And then suddenly there's one different one. So it just chopped off. But then you still have these fairly long clusters. And I was able to identify these long ones, and I can be pretty sure these are not individual clusters. They are just remnants from the repeated protein patterns in the genome. So I was able to eliminate them, and then found many more clusters. So I was pretty happy with that. Although I don't understand any of it, you see, I can only give it IDs. But during the analysis of this thing, I also take the data file or the annotation files. So each of these, this one, they have a data file which has DNA data, and they also have annotations. The annotations list, what they think these are proteins, okay, and they can identify certain proteins, and give it certain protein. All the known proteins have known names, and in the database they usually assign each unique protein with a different ID. So all this information are in the annotation file. And what I did was I would extract from the annotation file all the known proteins, where they start, where it ends. So I just put the starting position of a protein and ending position, and I insert all these things into this file. So I can see, the protein, and suddenly there's in between two proteins, there's some non-coding region, and then you can find some clusters in between proteins. So lots of the clusters are in between proteins, but lots of clusters are inside protein or occupy across the boundary of protein. So these are all things I don't understand. I think their analysis of their identification of proteins are problematic. Okay, and they will search their database and try to identify, and they may not correspond to real protein. I think for real protein, you really have to get all the proteins and get the sequence of proteins, and on protein sequence go back to the DNA sequence, and then do the comparison inside the genome. So just use computer programs to identify these proteins. I don't think these are useful, but you don't have to take this data as reliable. Okay, so I was able to look into all these things, and then so when I have a long list of this protein with inserted annotations, and I was able to identify the clusters. And generally, I use a big Excel file. So you have all the one column which has all the patterns, and whenever I found a cluster, I will just copy it into the neighboring columns, and this way, in the Excel file, you can look at the top 50 rows and see everything. Capture 10 or 12 of these clusters and look at them and try to make some sense of them. So this is where I do my analysis. And it is Excel is a very nice tool to handle all these the big data form, especially if we can formulate the data into the tab basis columns, and this way you can display them very conveniently in Excel and do comparisons and to seek some detailed understanding of it. Okay, so this is all very good. I think I don't know any microRNAs, but I can detect many of the unique 20 base patterns, and which forms clusters in their genome. So I think at this point I would be happy to see, we have microRNA-like patterns, and they do form clusters. Okay, that's all I can say about it. But then I was say, well, I have six bacteria, and so I have identify many unique 20-bit patterns. Can any of these 20-base pattern repeated across species? Okay. If they are microRNA, if they are basic instruction set of the cell computer, and if all the different organisms are built based on the same principle, and maybe we could find the same 20 base patterns across different species. Okay, so this was also quite easy to do. You collect all the bases, and then just attach them one to the next in Excel file, and just let the Excel to sort. And if you sort it, if each one, I think I gave this one, each one I gave them a different ID. This is R00001 to R305 to R00305, and this one is an N, and this one is G, and this is a Q, and this is, I think, an L, and this is a Y or something. So all these code are different code with unique letter identify them. And so if you have all this information in a sequence, it just tell you to search the not the sort everything according to the ID, and if they're not the same, they should have. Oh, no, I'm sorry. It's that in this file, I have two columns. I have the ID column and then the base. And this is actually I have ACGT bases. So I have these files which has a ID column and a base column. So I put all 6 of these files together and then search according to the base. If the same base would show up, okay. So in this way, I would search this one and look the IDs to see if they belong to different bacteria. So I'm getting everything set up and so, and it was very. I got 20 matches, okay. Among all these, I think I have a couple thousand, 2,000 or 3,000, 4,000 of these patterns, all these unique patterns, and I saw them and found that there are 20 bases which are common to several different bacteria, but they all started and they all have the same same starting patterns, and then 12 other things. That doesn't make anything useful. Okay. So I was very, very disappointed in that there are not more variable patterns which are shared among different bacteria. So that's what I found, okay? And I was kind of very disappointing in that I cannot, I can identify many unique 20 base patterns in the 6 bacteria, and I can identify many clusters in the genomes of these 6 different bacteria. But basically, these unique patterns that are found in each genome does not repeat among the different bacteria. So bacteria do not share the same 20 base patterns. So I don't know. That's a disappointment, but that's what I found. Yes.
By the way, this has been really inspirational. I got some comments after. But,
Yes.
A quick question. So, when you find the groups and the patterns, can you associate a behavior that those patterns trigger in the organism?
No. But people have found that in the microRNAs in the higher organism, they do have very specific. Some of them have very specific function. They can inhibit the produce of protein or enhance the produce of protein. So that's people have identified some of the functions in microRNA. But I believe the microRNA has a much more functionality than just inhibit or enhance the production of protein. I think that they are actually the control mechanism which produces RNA. And the RNA they produce can be can produce that list of microRNAs that's encoded in that piece of RNA, and this thing will go on doing other different things. Yeah. Yes. Mm-hmm. Yeah, I'm done.
Motion to adjourn is always in order.
Okay. Thank you very much. Okay, thank you.